Hello, sir. Hello. Hello, sir.
Yes, hello, hello, yeah.
Okay, sir. So, shall I start the session?
Okay, sure, no
Yeah,
problem.
great. So, good noon, guys, and everyone. Welcome to the session and a warm welcome to our guest speaker as well. I hope everyone is doing great and feeling energized. So, go ahead and grab your coffee or juice to stay refreshed. Let's keep this interactive and fun, and feel free to ask your questions anytime. So, ladies and gentlemen, and dear friends, a very warm welcome to all of you. It's truly a pleasure to have you here for today's exciting session. We are gathered to explore something both fascinating and meaningful, from Wasp Venom to Vision Rescue, exploring Phlototoxin 3434 glaucoma neuroprotection. The very title itself reminds us how nature's mysteries often hold the key to solving some of humanity's greatest challenges. Today's discussion is not just about science and medicine, but about hope, resilience, and the endless possibilities that innovation brings. Who would you have thought that something as intimidating as Wasp Venom could inspire a potential breakthrough in protecting vision? This is the beauty of research and turning challenges into solutions and nature's secrets into life saving opportunities. As we begin, I encourage you all to open your minds and ask your questions and engage deeply with the topic. And remember the words of Albert Einstein, in the middle of difficulty lies opportunity. Together, let's embrace the spirit of curiosity and discovery that drives us toward better health, brighter futures, and lasting impact. Before we begin Dr. Muhammad's session, I would like to take a moment to introduce him. So, alright, everyone, I hope you are ready for some serious inspiration because today we have the absolute honor of welcoming a truly remarkable personality to our session. So, please join me in giving a very warm welcome to Dr. Muhammad Fattah bin Fazel, Research Manager, Innovation and Commercialization, and lecturer at KPJ Healthcare University, Malaysia. With a PhD in pharmacy from University Kuala Lumpur and a strong academic background in chemical engineering technology and biomedical science, Dr. Fatak has built a career that beautifully blends deep academic knowledge with real world healthcare impact. What makes Dr. Muhammad so inspiring? It's not just his expertise in areas like neuropharmacology and molecular therapeutics and retinal neuroprotection, but also his unwavering commitment to education and innovation. He has played a key role in developing curriculum, guiding students at both undergraduate and postgraduate levels, and mentoring future biomedical scientists and pharmacists. Beyond HPJ, he also contributes as a part-time lecturer at UITM, extending his knowledge and passion for research and across multiple institutions. His ability to translate complex scientific research into commercially viable healthcare solutions shows as the power of bridging in science with innovation. Dr. Mohammad's research excellence has been recognized worldwide through the publications in top journals like Frontiers in Neuroscience, Frontiers in Pharmacology, and PLOS 1 and more. He has also served as a reviewer for international journals and has earned multiple awards for his groundbreaking contributions. Truly, his journey reminds us of the quote by Thomas Edison: genius is 1% inspiration and 99% perspiration. His dedication proves that great achievements come from hard work, vision, and the courage to innovate. And today we are privileged to learn from him and gain insights that can inspire us to push boundaries in our own field. Before we dive into today's session, I would like to take just a moment to introduce you to something that's close to our hearts: Thames Tech Solutions, a proud Swiss venture that's changing the way we see and use technology. At Thames, we stand by three simple but powerful values: innovation, excellence, and affordability. Whether it's cutting edge engineering or next gen scientific tools, we believe everyone should have access to transformative tech. As a visionary, Steve Jobs once said, innovation distinguishes between a leader and a follower. So, and at times, we choose to lead with purpose. From digital marketing and web design and AI powered tools, server management and mobile app development, our work spans across the industries like aerospace, medical applications, energy, and more. We don't believe in one size fits all. We believe in tailored solutions that help businesses thrive in a world that's constantly evolving. Actually, as Albert Einstein wisely said, strive is not to be a success, but rather to be of value. That's our mission. To add real value through strategy, technology, and a mindset focused on progress. But what makes us most proud is not just our business, it's our impact. With our initiative Mastering Up, we are focused on empowering the next generation by equipping students, especially from rural and underrepresented areas, with real world skills for real world success. Because as Nelson Mandela also said, education is the most powerful weapon which you can use to change the world. So today, as we begin this session, let's not just learn, let's imagine and build and shape a brighter tomorrow together. So the future starts now. So okay, at the end I would like to introduce to the motto of Dr. Muhammad, compassion in research and passion in discovery. So I would like to hand over the session to Dr. Muhammad, and also thanks a lot for joining us today, and over to you, sir.
Okay, thank you for the really nice introduction. I'm quite humbled by the introduction. Really, thank you. All right, so before we begin today, my name is Dr. Muhammad Fattah. Okay, I'm from Malaysia. I'm a research manager. Okay, before this, I've involved in quite a few of research. Okay, the part of it is this research. Okay, exploring philanthrotoxin 343 for glaucoma neuroprotection. Okay, so I'm using this kind of wasp venom. Okay, extract the wasp venom and then have the analog and whatnot, and I use it to check whether it can stop the progress of glaucoma or not. All right. Okay, so glaucoma it is not well discussed matter when it comes to blindness. Okay, people always talk about cataracts and people always talk about this short sightedness, long sightedness, all those kind of things, but people rarely talk about glaucoma. All right. So actually, glaucoma is the one leading cause of irreversible blindness globally. Okay, without we are knowing, it can cause it has caused irreversible blindness globally. All right. So to say that the prevalence is quite high and it is increasing more and more throughout the years. Okay, and we should be aware of this kind of disease, of this kind of neurodegeneration disease. All right. So it is characterized by the degenerative changes in the optic nerve and the retinal ganglion cells, okay, which is situated or located in our retina. Okay, so you must know that if there is no retinal ganglion cells, okay, our optic nerve can damage and it can cause our vision to be slowly deteriorating, all right, and leading to irreversible visual field loss. Right, so your vision will be, your peripheral vision will slowly become smaller. Okay, now you can see. Okay, you can see your peripheral vision can see up to here. Okay, but if you are having glaucoma, okay, it will become smaller until you cannot see anymore. Okay, that is how glaucoma do to your vision. Alright. So usually people will talk about elevated intraocular pressure. Okay, intraocular pressure is a pressure that will build up. Okay, that will build up inside your eyes. Okay, that will cause some kind of damage towards the optic nerve. Okay, what will happen is that the pressure will build up and it will push the optic nerve. Okay, this is like the back of your eyes. This is the optic nerve. Okay, the pressure will build up. Okay, will build up slowly, and will push the optic nerve. So when it push the optic nerve, it can cause some kind of um, it will induce some kind of apoptotic pathway. Okay, so when the apoptotic pathway is induced, it will cause retinal ganglion cell death. Okay, remember RGC, it is for our vision, it is for our vision, our how we see and whatnot. Okay, it will transfer the information from the retina to the optic nerve. All right. Okay, so if left untreated, it can result to permanent blindness. Okay, once glaucoma is progressing, you cannot stop it. So you have to take some kind of medication to lower the intraocular pressure. Okay, and sometimes there are some kind of mechanism or some kind of therapy that will decrease the intraocular pressure. Okay, um, by having some kind of stand or some kind of pipe. Okay, uh, they will plug into the iris and whatnot, and then it will lower the pressure of the eye. Okay, so moving on. Glaucomatous changes often progress despite IOP lowering and appear in patients even with normal IOP. Right, that's the problem nowadays. Okay, so even though there are some cases that IOP, the intraocular pressure, is lowering in the patient, but the progression of glaucoma still persists. Okay, so the glaucoma progression is still there. Okay, the field of vision, okay, the tunnel vision is continuing to develop and whatnot. Alright, so many patients continue to develop RGC loss, alright, regardless of a reduction of IOP. Okay, so um, okay, so you can see that the healthy eye or eye with the glaucoma and whatnot, alright? Um, okay, okay, you can see here, okay, there is a difference there. Okay, so this is a healthy eye, okay, where the optic nerve is good, the optic nerve is there and whatnot, okay? So, it's really alive and really healthy. Okay, so you can see here is very clear. Okay, but in some cases with glaucoma or in glaucoma, right, you can see that the optic nerve, okay, is start to die. Okay, it's dying and whatnot. Okay, what happened? Your eye will become cloudy like this. Okay, it will become cloudy like this. Right. So that's the difference between the healthy eyes and glaucoma. Okay, why it become like this? Because it is due to the optic nerve. Okay, optic nerve is slowly dying. Okay, so it can or it is differs by the cupping of the optic nerve. Okay, so the cupping of the optic nerve will cause the eyes will become like this. Okay, to become like this. All right. Okay, now for an NMDA receptor, okay, this receptor is kind of our target in the study. Okay, we want to check whether if this philanthrotoxin can block this NMDA receptor. Okay, and then if the blocking can cause any kind of progression or will stop the progression of the glaucoma itself. Alright, so before we design the study and whatnot, we already checked whether um, the pathway of the study, okay, the pathway of the research or whatnot. Okay, we demonstrated or previous study has demonstrated or we found out that increased activity of this receptor, okay, or this N-methyl D-aspartate receptor appears to be implicated in the pathogenesis of glaucoma. Right. So meaning that this NMDA receptor plays a pivotal role in this pathogenesis of glaucoma or the progression of the disease itself. Okay, how? Okay, so NMDA receptors are important in synaptic transmission and Um, in the essential categories, they have a few subunits, right? So our target is the NR2A subunit, okay, which is very abundant in the retinal ganglion cells, alright, in the retina. Okay, so we focus on this subunit, okay. So targeted NMDA receptor is likely to yield beneficial outcomes, and this outcome may in fact be dependent on the type of drug NMDA receptors interaction. Okay, so we want to use this theory, we want to use this idea, okay, of using this philantotoxin, okay, as a potential drug to block the NMDA receptor and causing the calcium influx that cause the glaucoma progressions to stop, right? Okay, so what happened is that our stimulation of an NMDA receptor has been associated with the increased intracellular calcium ion concentration. Okay, that's the idea. Okay, NMDA overstimulation, okay, will increase the intracellular calcium ion concentration. Okay, so when this calcium ion concentration increase, okay, it will lead into damage to the RGC and their axons on the optic nerve. Okay, so the idea is that when there is too much calcium coming in into the cells, okay, it will cause a series of pathways being activated. Okay, so when the pathway is activated, so what happens is that it will cause the cell to apoptosis. They will cause the cell to die, okay, which leads to the damage of the retinal ganglion cells and the axons in the optic nerve. Okay, that's the problem here. So our stimulation of NMDA, okay, can cause some kind of cellular death and whatnot. Okay, so we want to stop that. Okay. Okay, so how normal NMDA receptor normal physiology? Okay, so this NMDA receptor is being activated by glutamate. Okay, glutamate will bind to the NMDA receptor. Okay, thus opening the channel, making that calcium and sodium coming up into the cells. Okay, that is the normal physiology. Okay, glutamate will bind to the NMDA receptor. Okay, it will open the channel pore, and then after it opens, okay, calcium and sodium will come into the cells. Okay, these are the normal physiology. Okay, what happened is that in glaucoma, okay, we found out that glutamate is continuously activating the NMDA receptor. Okay, so when it activates the NMDA receptor continuously, what happens is that calcium will go inside into the cells, okay, in uncontrollably. So when it goes uncontrollably, what happens is that, like I said earlier, it will triggers the pathway. Okay, so what kind of pathway we are going to look into later. Alright, so in glaucoma pathophysiology, okay, the first thing first is the NMDA receptor overstimulation due to glutamate excitotoxicity. Okay, so when there is too much glutamate, okay, presence in the extracellular space, it will continuously stimulate the NMDA receptor. Okay, so when it happens that, okay, influx of calcium ions into the cytosol or into the cells, okay, can cause calcium overload that triggers the apoptotic pathway. Okay, so when it triggers the apoptotic pathway, and then it will triggers again. Okay, it's like a double effect. Okay, so it triggers apoptotic pathway, and then it comes in more, okay, and triggers more apoptotic pathway. Okay, as we all know, there is there is too many kinds of apoptotic pathway in our body, in our cells, right? Okay, so it becomes this double activation of triple and whatnot. Okay, it can cause optic nerve damage. Okay, so these are the summarized of the glaucoma pathophysiology. Okay, starting from overstimulation of NMDA receptor, okay, influx of calcium into the cytosols and whatnot. All right, and then it will trigger the apoptotic pathway and lead to optic nerve damage. All right, so RGC death, okay, retinal ganglion cell death. Yeah, commonly occurs due to overstimulation. Again, overstimulation of an NMDA receptor that can cause excitotoxicity. Okay, so excitotoxicity is when the cells is being excited too much. Okay, and it leads to the toxic condition. Right. So it associated with increased intracellular calcium ion concentration that culminates into damage to RGC and their axons in the optic nerve, eventually leads to glaucoma. Mm. So, prolonged excitotoxicity or prolonged overstimulation of the NMDA receptor can cause glaucoma. So we want to stop that. We want to stop this kind of overstimulation. We want to stop this kind of over excitotoxicity and whatnot. Okay, we want to block the receptor, and we don't want any more calcium coming into the cells. Okay, so by this idea, okay, we kind of stipulate lah. Okay, right. We kind of thinking that, oh, maybe we try to tackle this pathway first. Okay, we try to tackle this calcium regulated pathway first. Okay, in our study, right? So, what happened is that when there is increase in intracellular calcium ions, okay, inside the cells, okay, we have this calcium regulated protein. We have cabin 1, calcineurin, and also calpate. Okay, you must know that cabin 1 is bind to calcineurin, okay, in its inactivated form. Alright, this is inactivated form of calcineurin. Okay, so when it senses there's too much calcium inside the cells, what happens is that calpate, okay, will unbind cabin, okay, and calcineurin will become activated. Alright, so after calcineurin is activated, what happens is that it will stop the action of BCL2. Okay, what is BCL2? BCL2 is a anti-apoptotic protein, or is a protein that can stop the cell death. Okay, so it regulated the cell death. Okay, it guards the cell from dying. Alright, so what happens is that calcineurin will stop the action of BCL2 and it will induce the action of BAX. Alright, so BAX is a protein which is inducing the apoptosis. Okay, it will trigger the apoptotic process in the cells. Alright, so after a series of pathways, after a series of what we call it, after a series of pathways and combinations and activation and cancellations and whatnot, okay, what happens is that mitochondria will start to burst out the cytochrome C and whatnot, and then beginning the cascade of apoptosis, okay, and then we have this caspase 3, which is like very fundamental protein for apoptosis. Okay, if you have this protein inside your cells, inside your tissue, it means that your cell is undergoing apoptosis or cell death. Right? So what will happen is that it will cause changes in behavioral pattern and visual ability. Okay, changes in retinal and optic nerve morphology changes. Okay, so this is our focus for today. Okay, on the 2. All right. Behavioral pattern and visual ability, and also changes in retinal and optic nerve changes. Okay, so that's all lah. Alright, so okay, so as I said earlier, glaucoma is a set of disease, okay, a group of diseases that can cause damage in the eyes optic nerve. Okay, it will damage the optic nerve. Alright, so a healthy optic nerve is necessary for good vision. Okay, of course, if you don't have your optic nerve, you cannot see. Okay, if your optic nerve is damaged and whatnot, your vision will be impaired. Okay, because there is no connection between your eyes and your brain. Alright, so when pressure increase the optic nerve, glaucoma can develop. Okay, so the original idea is that we can say that IOP increase, or the intraocular pressure increase inside the eyes. Okay, glaucoma will develop. Okay, so most of the focus of current treatment is decreasing the IOP. Okay, so most of the current treatment nowadays, okay, they only focus on lowering the pressure, lowering the intraocular pressure. Alright, so Not every person, but okay, not every person with increased eye pressure will develop glaucoma. Okay, as I said earlier, not all those even though the intraocular pressure is lowering, but glaucoma still persists. Right. So low tension glaucoma, okay, developed without increase in the eye pressure. Okay, so in this study, we focus on the low tension glaucoma and also called as normal tension glaucoma. Alright, so this is our problem statement lah. Okay, problem with current treatment. Okay, usually it's very expensive to get all those medications. Okay, to get all those therapy. Alright, and then only have suboptimal IOP control. Okay, meaning that suboptimal IOP control. You consume the drug. Okay, you apply the eye drop in your eyes and whatnot. But the IOP control is very low. Sometimes patient does not react to the treatment. Okay, so there is problem with current treatment. Okay, then we have this adverse reaction with ocular allergy. Okay, some patient have some kind of allergy towards the drugs and whatnot. Okay, we have bradycardia because we are using beta blocker. Okay, for the treatment such as timolol. Okay, so we are using beta blocker as the treatment and whatnot. It can cause bradycardia. Okay, your heartbeat will become what we call it, um, slower and whatnot. Okay, and we have irregular pulse. Okay, it can cause some kind of disruption in your vascular system. Okay, so when we have this, the treatment may not suitable with people that have vascular disease such as heart attack, okay, such as heart problems, such as high blood pressure and whatnot. Okay, they are not suitable for this kind of disease. Okay, so hence there is a need for a new therapy, okay, to address retinal ganglion cell death while providing simultaneous IOP reduction. Okay, so this is what we want to tackle in this problem. Okay, we want to address directly, okay, we want to stop directly the retinal ganglion cell death by the neuroprotection of pilantotoxin. Alright, okay, so what is pilantotoxin? Okay, pilantotoxin is wasp venom. Okay, wasp venom isolated from a solitary beagle wasp. Okay, this is the wasp. Okay, Philanthus triangulum. Okay, so actually we isolated original compound, which is 433. Okay, so 433 here, this is the original compound. Okay, but to ease the analog reproduction, okay, we want to make some kind of large scale production of this venom, of this compound. Okay, so we switch the polyamine tail. Okay, we switch the tail. Okay, so we have this 433. Okay, 1 2 3 4. Okay, 1 2 3. So we switch. Okay, so by switching this, the synthesize will become much more simpler. Okay, and much more parallel and symmetry. Okay, 3 4 3 1 2 3 4. Alright, so this is the analog that we come up with, the or the analog that my collaborator come up, okay, from the 433. Alright. Okay, even though we have changed the structure and whatnot, okay, what happened is that it retains pharmacologically active properties of the original compound. Okay, it does not change anything. The effect does not change. Okay, it's still the same. Alright. So it is a non-competitive open channel blocker for NMDA receptor and act as a basis of its neuroprotective effect. Okay, so with this kind of idea that we come up, okay, so we make it as a basis for the protective effects. Okay, why? Because it is structurally large hydrophobic head and positively charged tail. Okay, so what happens is that if you have this large hydrophobic head and positively charged tail, okay, large hydrophobic head, the head is big, all right, so, and it is hydrophobic. Okay, what happens is that it will stuck into the channel pore, okay, and into the channel pore correctly, and then it is positively charged. Okay, so the cell will be you must remember that calcium ion has the positively charged, right? Okay, it's a positively charged. So what happens is that this positively charged tail will replace calcium. Okay, it will replace calcium from going into the cells. Instead, the compound will bind into the receptor itself. Okay, so it can bind this within the channel pore. Okay, that is the idea. This is the main idea why we are choosing this phenanthotoxin 343. Okay, so these are the blockade of the NMDA receptor by PHTX. Okay, what happens is that it's quite fast, right? Okay, so when glutamate binds into the receptor, okay, PHTX will go into the channel pore and it will prevent sodium and calcium from going into the cells. Okay, so this is the mechanism, or this is the
We're now
PHTX
session
blockade
yarkana
on the
முடிச்சிட்டு
NMDA
கூப்பிடுறேன் சரியா.
receptor.
Bye bye.
Okay, so moving on, okay, enough about the introductions and whatnot. What is phenanthotoxin? What is NMDA? What is glaucoma and whatnot? So we move on to the study itself, right? So, general objective of this study, okay, to determine the neuroprotective effects of molecular mechanism of PHTX343 against NMDA-induced retinal and optic nerve injury in rats. Okay, so we are going to look into the neuroprotective effects, okay, of PHTX343 and against NMDA-induced retinal and optic nerve injury in rats. So we induce the NMDA into the rats. So we induce the NMDA excitotoxicity in the rats by injecting NMDA itself into the eye of the rats. Okay. So for your information, all this data in this slide has been published in PLOS One, okay, a Q1 journal. All right, you can look at it. Entitled "Phenanthotoxin 243 Attenuates Retinal and Optic Nerve Injury." Okay, protect visual function in rats and NMDA induced excitotoxicity. All right, so moving on. Okay, the study design. All right. So when we're talking about study design, it's quite challenging to figure out what kind of things that we want to do, what kind of things that we want to test. All right. So the things that we do is that for all the experiments, all right. So we divide the rats into three groups. Okay, the first one is PBS or polyphosphate buffer saline group. Okay, and then NMDA group and also PHTX 343 group. Okay, so there are people wondering, okay, why we don't compare with the drug, with the existing drug and whatnot. Okay, the answer is that there is no marketable drug or approved drug for glaucoma in tackling the neuroprotection side. Okay, so we cannot compare with the commercially available drug because there is no drug that tackle the neuroprotective effect of the glaucoma. All right, so before we inject the before we move on with the other parameters and whatnot. Okay, so we have this three group and then intravitreal injection with NMDA and whatnot on the treatment group. Okay, then we have after 7 days of treatment, we euthanize, okay, the rats, and we look at the retinal morphology and also optic nerve morphology of the retina and optic nerve, and then we move on to the behavioral assessment, which is locomotor activity using open field test and also open field arena. Alright. Okay, the first thing first, we are going to look at the behavioral assessment. Alright, so the behavioral assessment were divided into two. Okay, we have this open field test and then we have this object recognition test. Alright, so but why we do this? Because we want to check whether page 343, okay, will have some kind of effect towards the behavioral analysis. Okay, so we want to check whether the vision, okay, being impaired by the NMDA, okay, can cause some kind of blindness to the rats, okay, or can cause some kind of what we call it, we will able to recognize the object, or can check the total distance travels and whatnot, okay, through this open field and object recognition test, right? Okay, so we have for open field, we have total distance travel, and then we have total immobile time, and also we have total immobile episodes. Okay, these are the three parameters in the open field test, right? And then we have this object recognition test, all right, we have familiarization, we have object displacement, and also we have object replacement test. Okay, so these are the design for the visual behavior analysis, okay, that I did, okay, back then. Okay, so for open field test, we have a trial for 10 minutes and then familiarization stage, okay, where the rats are being familiarized with all the object in the open arena, right? And then we have object displacement, okay, where we displace a few objects into a different location. As you can see here, alright, this object has been moved up here, okay, this object has been moved between these, okay, object A and B and whatnot, okay, and then we replace the object, okay, we replace some of the object, which is object A1, okay, we call this object A1, right, we replace with object A, okay, so this is object A and replace with object A1, okay, these are the study design, alright, so it's the same lah, okay, it's the same with the previous one, alright, so A, B, C, D, E, and then for familiarization, and then we move the object D to here and object E to up here, and then we replace object A to object A1. Alright. Okay. So for visual behavior analysis, the result is kind of tricky. Okay, the result is kind of hard to what we call it, hard to think about, how hard to explain and whatnot. Okay, so bear with me for a while because this is going to be a bit confusing. Alright, so but don't worry, I will summarize all the result and tell you what it's all about. No worry. Right. Okay, so for the first one, the first parameter will be total distance travel in open field test, okay, with object replacement and displacement. Okay, alright, so there is too much graph here, right? So what it says is that no significant changes were observed between group in trial 1. So trial 1 is a habituation phase, meaning that we try to give the rats a bit of time, a bit of space to explore, right? So all those group will show the same time, okay, the same distance travel, okay, because they just want to explore the area. Okay, that is for open field and habituation stage. Alright, and then we have NMDA group recorded the highest total distance travel across trial 2 to 4. Okay, so what happened is that here, okay, NMDA group, even though it has been introduced or it has been in the habituation stage, okay, and it been reintroduced into the same situation or its same environment, it's still discovering, it's still the total distance travel is still high, okay, because indicating that the visual, that their visual is not good. Okay, they have, every time they are being introduced into the new environment or into the same environment they will continue to explore further, okay, to explore every kind of object and whatnot that is placed in the arena. Okay, so you can see here, okay, 2, 3, 4 still high, okay, and then we can see that total distance travel by rats in the PHTX group here and also PBS group is shorter, okay, because it indicated that they can see, okay, PBS is normal group, they can see well, and PHTX 343 group is the treated group, okay, meaning that the treatment is working, okay, it's almost working for the rats, okay, as you can see here, right, across trial 2 to 7, Okay, it is comparable with PBS, okay, with P more than 0.05. Alright, okay. Okay, so next we move on to the total immobile time. Okay, it contradicts with the total distance traveled. Right? So you can see NMDA group recorded the lowest total immobile time across all trials. Okay, what happened? Because NMDA group did not see well, cannot see well. So what happened? They kept exploring the arena. Okay, so they kept exploring the arena. So their immobile time, okay, the time they stop is very short. Okay, so because they are always on the move exploring the area. Okay, but for PHTX 343 and PPS, okay, we can see that the total immobile time is significantly greater, okay, trial across trial 2 to 7. Okay, so this is what happened when the visual ability of the rats is being impaired. Okay, and if the visual ability of the rat being is being treated well, okay, by our compound. Okay, next we have total immobile episodes. Okay, so NMDA group recorded the lowest total immobile episode across all trial. Okay, it reflect with the total immobile time. Okay, so this total immobile episode, how many time the rats stop. Okay, as you can see here, NMDA did not stop too many times. Okay, so they keep on walking, continue to explore the arena, right? And also PSTS group and PBS group is comparable across trial 1 to 7. Okay, so I hope you can see the difference between the total distance travel, the total immobile time, and also total immobile episode. Okay, it just want to show you how the rat explore the arena if their vision is being impaired or their vision is being treated well by the compound. Okay, next we have the familiarization phase. Okay, so for familiarization phase, okay, all rats are being familiarized in the arena. Okay, all rats are being familiarized with the object and whatnot. So there is no significant difference among all group during familiarization phase. Okay, all rats showed similar pattern in exploring the arena. As you can see here, okay, the pattern is similar. Okay, the rats are exploring. Okay, the rats are exploring the arena, exploring each object and whatnot. Okay, all the same. Okay, and then for object displacement test, if you remember, we displace the object. Okay, object D to here, object E up here and whatnot, right? So what happened is that NMDA group spent significantly more time at the original object ABC. Here, this is NMDA. Okay, okay, object A, B, and C. Okay, they stay at the same object. Okay, so they spend significantly more time on the original object ABC compared to PHTech 343 and PBS. Okay, and then PHTech 343 and PBS group react in total opposite. Okay, they only explore the displaced object only. Okay, object D and object E. Okay, for PBS and PHTech 343 group. Right? That's the difference. Okay, an MDA will explore all the object, but page text and 343 only explore the object D and E, the displaced object. Okay, next, object replacement test. Okay, where we only we change object A to object A1, right, like before. Okay, so an MDA group explore all objects with nearly equal frequency again. Okay, they don't care about the new object. They continue to explore all objects. Okay, because the vision they cannot see. Okay, they cannot see properly. Okay, this object they already put there for a very long time and whatnot. Okay, and for object A1, okay, for PHTX and PBS group, you can see here. Okay, they only explore for PBS and PHTS group, sorry, okay, they only explore object A1. Okay, while the rest they are not frequently visited lah. Okay, object A, object B to object E, because they are the same. Okay, because they already see the object, so they don't want to go to the current object. They only go to the newly introduced object. Alright. So that's the difference. Okay, replacement, displacement, and also the familiarization test. Alright. So as a summary of all that thing, right? It was observed that NMDA illustrate explore the arena more compared to control and pace text 343 group. Okay, it is possibly due to their inability to see like a normal rat. Okay, and they need to learn each time they were introduced in the same or new environment with different object shape reconfiguration. Alright, so because NMDA rats is have the inability to see like a normal rats, okay, and they need to learn each time they are being introduced into the same configuration, okay, or the new environment or the same configuration of object, right? So, page text 343 treated right recognize the environment possibly due to better vision, right? Hence, explore the arena considerably less compared to an NMDA rats and comparable with healthy rats or the PBS rats, right? So, what we conclude is that this shows page text 343 pre-treatment, okay, prior to NMDA induction, protect against visual impairments caused by NMDA induced retinal and optic nerve injury. Okay, this is for the visual behavioral analysis part. Right. Next, we move on to the retinal morphology assessment and also optic nerve morphology assessment. Okay, both by H&E staining or hematoxylin and eosine staining and also collagen blue staining. Okay, so the objective, okay, again, almost the same, okay, effect of PSTX against retinal morphology using H&E. And effect of page 343 against induced and then they induce optic damage in rats using collagen staining. Okay, so we are looking at the retina and we are looking at the optic nerve, both things, right? Okay, so as you can see here, okay, so for the arrow, okay, you can see here arrow. This arrow indicate that there are the presence of retinal cell nuclei. Okay, we cannot say that this is a retinal ganglion cell because we didn't do the specific staining for the retinal ganglion cell. So what we can conclude here is that there are retinal cells nuclei in the retina. Okay, because we are only using hematoxylin and eosine staining. Okay, so as you can see here, there are the retinal cells nuclei in the PBS group is almost the same. Okay, the numbers is almost the same with the PHTX 343 group. Okay, but we can see here in the NMDA group, okay, there are only 1, 2, 3, or maybe 4. Okay, so the comparison between PHTX and PBS group is almost the same. Okay, indicating that the PHTX 343 group is protecting the retina from the excitotoxicity. Alright. So not only we do the what we call it qualitative analysis, okay, by the staining. Alright. We also do the quantitative analysis. Okay, what we do is that PHTX 343, okay, increase the numeric retinal cells density. Okay, the density of the retinal cells, okay, in the ganglion cell layer. Okay, this is the ganglion cell layer up here. It retains the numbers of retinal cells density. Okay, and also the retains linear retinal cell density also. Okay, it is comparable with the PBS as you can see in the graph here. Okay, comparable, but NMDA is very low. Alright. Okay, next we move on to the optic nerve grading. Okay, optic nerve grading, we have grade 1 until grade 4. Okay, adapted from my seniors. Okay, so for grade 1, normal axons. Okay, then we have grade 2, we have grade 3. The degenerations are starting to appear and whatnot, right? And then grade 4, all axons showing degeneration. Okay, you can see here all those empty space and whatnot. That is called degeneration. Alright. Okay, so again, it reflects with the HNA staining. Okay, as you can see here, PBS group, okay, is almost similar, have almost similar morphology with PHTS refractory group, okay, while NMDA has this kind of extensive clearing of nuclear, okay, or we call it vacuolization, okay, vacuolization meaning that this layer or this morphology, okay, is being replaced with a fibrous tissue or fatty tissue, which is meaning that they are not functioning anymore. Alright, so again we use the quantitative method. Okay, we check the grade, we calculate the grading and whatnot. Okay, we can see that NMBA has the most what we call it, most degradation of the optic nerve. Alright, so you can see page tag there is degradation, but it is not as much as an NMDA, and it is almost comparable with the PBS group. Okay, almost comparable. Okay, so as a discussion, glaucoma is an ocular pathology that characterized by the loss of RGC and optic nerve damage leading to blindness. Okay, from a therapeutic standpoint, NMDA receptors are potential targets for treatment of neurodegenerative disease, including prevention of RGC death and optic nerve damage in glaucoma. In the present study, or our study, demonstrate that pre-treatment with PHTX343 prior to NMDA registration result in reduced retinal cell loss compared to the NMDA treated group. Okay, so PHTX343 managed to reduce retinal cell loss in the NMDA treated group. Right? So, in accordance with the observations made by on the retinal morphology, okay, pre-treatment with PHTX343 resulted in lesser degenerative changes in the optic nerve. Okay, so what we can summarize is that our finding, both object displacement and replacement tests indicate that common trend that NMDA treated rat explored all the object. Okay, this is for visual behavior analysis. Okay, NMDA rat treated rat, okay, explored all the object with nearly equal frequency. Okay, while page that 343 explore the novel object with higher frequency as compared to the original object. Okay, so this is what have been summarized before. Okay, and NMDA explore all the object. Okay, increase the total distance travel, decrease the total immobile time and whatnot. But page that 343 managed to we call it decrease the total distance travel. Okay, and then uh, increase the total immobile time, okay, total immobile episode and whatnot. Okay. Alright, so as a conclusion, our data strongly suggests that this PHTX343 protects against NMDA-induced and cytotoxic injuries to the red retina. This was evidenced by the prevention of NMDA-induced changes, okay, in retinal and optic morphology by vision-dependent behavior of the rats. Okay, this is my our conclusion on the study. Right, so PHTX343 managed to protect, okay, the retinal cells and optic nerve from being injured further by the NMDA cytotoxicity. Okay, so we managed to stop the progression, okay, of glaucoma in the rats. All right, so what is happening right now? So what is after this project? Okay, so what we are doing now is that we are looking in the molecular aspect of neuroprotection. Okay, we are investigating the calcium regulated proteins. Okay, the calpain, the calcineurin, the cabin 1 levels. Okay, in the rats. Okay, we are checking the apoptotic protein. Okay, pro-apoptotic, beta caspase-3, and also BCL2, anti-apoptotic and whatnot. And then we are looking at the suppression of oxidative stress. Okay, by measuring the level of GSH, SOD, and also CAT. Okay, catalyst and whatnot lah. Okay, support dismutase. Okay, and then the latest one, okay, I've been studying on the molecular docking in silico model. Okay, what we do is that what my collaborator do is that they check whether this PHTX compound, okay, by predicting the potency and specificity of the SH343 towards the receptors subunit. So why we do this? Because we want to check whether this PSH343 can contribute to other kind of neuroprotection. Okay, for example, dementia, Alzheimer, Parkinson, and whatnot. So we are looking into that area also, not just the glaucoma. Alright, so in hope that we can further this study for a clinical trials and one in the future. Okay, make it into a standard therapy in neuroprotection. Alright, so I would like to acknowledge, okay, my team. Okay, we have my supervisor here, Dr. Izzuddin, and my colleague, Dr. Haikal. Okay, my collaborator, Dr. Reno, Dr. Igor, okay, Dr. Ian, and Dr. Hendrik, and also these two co-supervisors, Dr. Nurul Alima and also Dr. Nurul Hafiza, and the funding for this project as well, the FRGS and UniKL, Excellent Research Green, and also Energy Solution. Alright. Okay, thank you. That's all from me.
Yeah, thank you, sir. Thank you so much for the wonderful session. So, okay, sir, we have some questions. So, shall I ask now?
Yeah, sure.
Okay. So, in our first question, how do you see PSTX343 evolving from laboratory research into large-scale clinical applications for glaucoma patients?
Okay, thank you for the question. As I said earlier, we are progressing the research. Okay, as for now, we are still in the if you are familiar with the technological readiness level, we are approaching TRL 5. Okay, because we have all this validation and all this kind of result publications and whatnot that as a proof that this philanthrotoxin 343 is can be used as a what we call it, can be used as a treatment. Okay, can be as a candidate for the treatment for glaucoma. So as we approaching the TRL 5, okay, we are looking for a potential collaborator from industry and whatnot. Okay, so that is how we see this compound can go, how far it can go. Okay, so I think that's my answer to the question.
Yeah, thank you, sir. What future challenges might arise in ensuring the safety and long-term effectiveness of a venom-derived compound in human treatments?
Okay, so this has been like a rising matter. Okay, because when people say that, oh, venom, it can cause this kind of effect and whatnot, right? So actually, you must understand that venom is like this huge structure. Okay, we have this huge structure of venom that can cause many kinds of effect. Okay, so what we do is that we extract only tiny bit of the venom. Okay, only tiny bit of the venom, and then which is the philanthrotoxin 433. Okay, so when we extract the venom, when we extract the 433 from the original venom, and we synthesize. Okay, synthesize meaning that it is not original anymore. It's not from the original venom. We create an artificial analog. Okay, that has some kind of the same pharmacological effect. Okay, that can bind to the receptors well. Okay, as the same with the original compound. So meaning that for the concerns about the venom, about the toxins and whatnot, it should not be arising. Okay, plus we have done toxicity testing and whatnot, and we can see that the kidney, the liver of the rats is not being affected as well. Okay, because we only using PBS, okay, polyphosphate buffer okay, to dilute the philanthrotoxin. Okay, we does not use any kind of solution such as alcohol or xylene or whatnot. No, just a normal saline. Okay, so it should not affect the organ. It should not provide any kind of toxicity towards the towards the rest for now. Okay. For now lah, I'm not sure if we are progressing further, we can find any kind of other effects and whatnot. Okay, that is what I can assure you for now. Right.
Oh, thank you, sir. Sir, could PHTX 343 potentially be adapted to product neurons in other neurodegenerative diseases beyond glaucoma?
Yeah, currently, except from glaucoma, I'm assessing the effect of beta-toxin 343 on Alzheimer. Okay, so what happened is that I inject directly the philantotoxin into the hippocampus, okay, to see whether it can protect the brain from degenerating or not. Okay, so maybe it can prevent the neuron degeneration. Okay, it can prevent lots and lots of amyloid plaque building up and whatnot. Okay, so that is what I am currently trying to do, trying to explore, okay, with the basis of the neuroprotection of philantotoxin 343.
Okay. How might the advancements in nanotechnology or drug delivery systems improve the precision and effectiveness of PHTTX 343 therapy?
Okay, interesting question because nowadays people are going into nanomedicine, precision medicine, or whatnot, right? Again, moving forward, okay, I'm thinking of using philanthrotoxin 343. Okay, I'm going to attach it into a we call it some kind of nano precision vehicle. Okay, for example, iron or for example copper and whatnot. Okay, I'm going to use that idea to expand and look at the efficacy. It should be the efficacy and the potency of the compound will be much more higher. Okay, because it's a targeted, it's a precision medicine, right? Okay, so it's a targeted therapy. So it should go, go straight to the source. Okay, so if we want it to go to the eye, okay, we can direct it, okay, by using magnets and whatnot. Okay, so we can direct the medicine or the nanotechnology and whatnot towards the eye itself. Okay, so there are many possibilities that we can do with this compound actually. So because it's very new and people are not discovering much on the potential of philanthrotoxin 343. In fact, I am the first in the world to check this philanthrotoxin 343 for glaucoma. Okay, before this, no one has ever done this. Okay, so when we done this, we look at the what we call the neuroprotection, NMDA receptor interaction and whatnot. It is very, very good actually. So remember that I'm doing the further work on the molecular part and whatnot. The result is very promising and whatnot. Okay, so yeah, I think the possibility for this compound to go further is very, very high, but need to maybe need more collaboration and more ideas behind the study as well, right?
Oh, thank you, sir. Okay, so the final question. What role do you envision for global collaborations between researchers, clinicians, and pharmaceutical companies in bringing such novel treatments to patients?
Um, what am I envision? Okay, so actually our collaboration for this project is quite huge actually. So we have collaboration from Denmark, okay, Copenhagen, and we have collaboration from UK, okay, Nottingham. So what we do is that we always try to search for a new opportunity, a new vision, all right, to embark on some kind of um, what we call it, introduction to the pharmaceutical industry. Okay, in fact, right now I have filed a patent on the application for the philantotoxin for glaucoma. Right, So in hope that if the patent filing is done well, we can sell this idea to the pharmaceutical company or not. Okay, so yeah, as I said earlier, this thing can be further. Okay, the study can be further until clinical trials and whatnot beyond the animal study. Right. So there's an answer for the question.
Okay, great. So, thank you for the useful answers. They have been really helpful in clarifying the topic. I hope the participants were able to follow along and understand the key points shared. So, your insights have certainly value to the session. So, ladies and gentlemen, as we come to the close of this insightful session, it's my privilege to extend a heartfelt vote of thanks to Dr. Muhammad Fattah bin Fazil for sharing his valuable knowledge and inspiring thoughts with us today. His talk has
Okay. Good.
given us a deeper understanding of the fascinating intersection of science, innovation, and healthcare. And we are truly grateful for the time and effort he has dedicated to this session. So, Dr. Mohammad's passion for research, education, and innovation has left us all motivated to think beyond the boundaries and explore new possibilities in our own field. The way he bridged the complex scientific concepts with practical applications was both enlightening and thought provoking. His journey and contributions are a reminder of how dedication, curiosity, and compassion can drive meaningful impact in both academia and healthcare. So, on behalf of everyone present here, I sincerely thank Dr. Mohammad for his presence and for making this session memorable. We also extend our gratitude to the organizers and participants and supporters who made this event possible. As we conclude, let us carry forward the inspiration and insight shared today and continue striving for excellence in our endeavors. So, thank you once again, Dr. Mohammad, for being with us. It has truly been an honor. Thank you, sir. Thank you so
Thank you.
much.
Thank you for inviting. Thank you for inviting me. Right.
Great,
Can
sir.
invite me again.
Definitely,
I
we
don't
will
mind.
meet in future collaborations also.
Sure, no problem. I'm open. No worry.
Yeah, great. Thank you, sir. Take care. Bye
All
bye.
right. Bye.
Yeah, bye bye.